undergo changes, sometimes these chromosomes split, which is called a fission. So here we see a chromosome that undergoes a breakage event. In this instance, the resulting fragments, one of them retains the centromere from the previous chromosome. However, this new fragment does not have a centromere. And for this fragment to be retained in cells, it has a new centromere form, and those are called a neocentromere. This fragment does not contain repetitive sequence at that centromere because it is a new centromere. As time goes on, these neocentromeres gain centromeric repeats, and they become mature centromeres. So this process is called the maturing of the centromere. When chromosomes fuse, you will wind up with this chromosome that has two centromeres. These are called dicentric chromosomes. And this can cause problems in the cell cycle because you will have microtubules attached at this centromere and that centromere. And in some cases, it winds up shearing the chromosome. Over time, one of the centromeres is usually inactivated, so this guy here, which means that instead of having microtubules attached to both of these regions, they will only attach to one of the centromeres. So in effect, these chromosomes become more normal. There are examples where centromeres resurrect themselves, and one of the most famous examples is human chromosome 15. So in human chromosome 15, so the ancestral chromosome that contains human chromosome 14 and 15 results from a fission of the ancestral chromosome. So in effect, the ancestral chromosome undergoes a fission, and in the human ancestor, the chromosome 15 undergoes a centromere shift. At this centromere, there is or at the ancestral centromere, there are repetitive sequence. And if you think about a mature ancestral chromosome, this makes sense, that there would be centromeric repeats present. And this region is actually very active in human neocentromere formation. In addition, it still retains these centromeric repeats. So in human chromosome 15, there is an overlap of repetitive sequence, an ancestral centromere, and also it's a center of human neocentromeres. So in derivative chromosomes originating from human chromosome 15, you'll see this overlap. So there's something inherently marked, likely as an epigenetic mark. However, in the field it has not been identified yet. So we're very interested in these centromerization patterns.